Mrs. H here. We are going to learn how an electrical impulse or action potential is generated and propagated along a neuron and how myelination helps to speed up conduction. Here are three types of neuron: the sensory, the relay and the motor neuron. I'm just going to label the relevant parts for today's video. The relay neuron is easy to spot, and the main ways to tell the sensory and motor neuron apart are that sensory neurons are connected to receptors and their cell bodies are in the middle of a dendron and axon sticking up, as you can see here. A motor neuron's cell body is at one end and it has no dendron, only an axon. The dendron and axon are essentially the same structure, but the axons take impulses or action potentials away from the cell body. These are myelinated neurons, which mean that individual cells called Schwann cells are wrapped around the dendron and axon, making up a structure called a myelin sheath. The myelin sheath is a fatty layer, so wherever there is myelin sheath, ions can't move across the axon or the dendron membrane. Ions can only move across the gaps in the myelin sheath, which are called the nodes of Ranvier. Here we are zooming in to take a closer look at the structure of the dendron or axon membrane. You can see that the membrane has a phospholipid bilayer, voltage-gated protein channels, which are opened by a change in charge, and a sodium and potassium ion pump. The blue channels represent voltage-gated sodium ion channels and the red are voltage-gated potassium ion channels. The diagram shows the orange area as being outside of the membrane and the blue area as inside the dendron or axon. The sodium and potassium ion pump actively pumps out 3 sodium ions and 2 potassium ions into the dendron or axon. Because 3 positive ions are being pumped out and only 2 in, makes the inside more negatively charged compared to the outside, which is the same as the outside being more positively charged compared to the inside. The voltage-gated potassium ion channels are slightly leaky, so some potassium ions leak out, and this further increases the negativity of the inside compared to the outside. This particular movement of the ions happens when the neuron is at rest, and the potential difference across the membrane is around -60 millivolts and is called the resting potential. The membrane is polarized. Resting potential values can differ, but generally tend to be around -60 to -70 millivolts. A stimulus will cause a few voltage-gated sodium ion channels to open. This will mean that sodium ions can diffuse through into the dendron. And because they have a positive charge with them, the inside of the dendron will become less negative, which is the same as more positive. So it will go from -60 millivolts, -59, 58, 57, 56, 55, 54, 53, 52, 51, 50. If a threshold value of -50 millivolts is reached, then this is enough charge to cause other nearby voltage-gated sodium ion channels to open. As more voltage-gated sodium channels open, more sodium ions can diffuse into the dendron and will cause it to become even more positive. The inside will reach a value of +40 millivolts, which is an action potential. When the inside becomes more positive, we call this depolarization. The sodium ions will diffuse along the dendron or axon, and depolarization will cause subsequent voltage-gated sodium ion channels to open, which leads to more sodium ions diffusing in and another action potential. And these sodium ions will then diffuse along the dendron or axon, causing more voltage-gated sodium ion channels to open, causing another action potential. And this goes on and so on, all the way to the end of the sensory neuron until it gets to the synapse. So more sodium ions cause more voltage-gated sodium ion channels to open, allowing more sodium ions to diffuse into the dendron or axon. This keeps happening again and again. This is an example of positive feedback. Anyway, meanwhile, after an action potential has occurred, the voltage-gated sodium channels close and the voltage-gated potassium ion channels open, causing many potassium ions to diffuse out of the dendron or axon. That is going to leave the inside more negative, and this is called repolarization. The voltage-gated potassium ion channels remain open for slightly longer, which causes the inside of the dendron or axon to become temporarily more negatively charged than the resting potential. And this is called hyperpolarization. The voltage-gated potassium ion channels close, and the sodium and potassium ion pump will quickly restore the resting potential so that the neuron is ready for stimulation again if needed. The series of depolarizations or action potentials followed by the repolarizations will move along the whole neuron until the action potential reaches the synapse. This graph shows the change in potential difference across the neuron membrane. When the neuron is at rest, the resting potential is around -60 millivolts due to the sodium and potassium pump, pumping out 3 sodium ions to every 2 potassium ions in, and that some of these potassium ions leak back out, making the inside more negative. When a stimulus arrives, some voltage-gated sodium channels open and some sodium ions diffuse in, causing the neuron to become more positive or less negative on the inside, and around -50 millivolts is the threshold potential. If threshold potential is reached, more voltage-gated sodium channels will open, causing more sodium ions to diffuse in, causing further depolarization, and an action potential is reached at +40 millivolts. Notice that if threshold potential is not reached, then an action potential will not be generated because there needs to be enough of an increase in positive charge to cause subsequent voltage-gated sodium channels to open. The next part of the graph shows a decrease in positive charge called repolarization, and this is caused by the voltage-gated potassium channels opening and the potassium ions diffusing out. And this part of the graph shows that the charge is more negative than the resting potential. So this is hyperpolarization. And remember, that is caused by this potassium ion overshoot. Hyperpolarization means that threshold potential definitely can't be reached and the voltage-gated sodium channels will not be able to open. The period of time that the voltage-gated sodium channels do not open is called the refractory period, and this ensures that action potentials are transmitted in one direction, and it also enables the neuron to have a short period of time of rest before being stimulated again. Finally, when the voltage-gated potassium ion channels close, the sodium and potassium pump can restore the resting potential back to -60 millivolts. Remember what a myelinated neuron looks like, and that the myelin sheath prevents any movement of ions between the inside and outside of the neuron. The only place where ion movement can occur is at the gaps in the myelin sheath called the nodes of Ranvier. So the only place where action potentials can occur is at the nodes of Ranvier. The insulating effect of the myelin sheath ensures a steep concentration gradient of sodium ions at the node of Ranvier, meaning the sodium ions can diffuse faster along the inside of the neuron, reaching the next node of Ranvier quickly to cause voltage-gated sodium channels at this node of Ranvier to open, and so on and so on. This is much faster than in unmyelinated neuron because there are fewer ion exchanges along the whole length of the neuron. We're only getting exchanges at the nodes of Ranvier. Action potentials occurring at the nodes of Ranvier is called saltatory conduction, So called because the action potentials appear to jump from one node to the next node inside the neuron. And that is it. If you found this video useful, please like and subscribe for more content and check out biologybreakdown.co.uk.